Hello, chemistry students. Today we're going to be performing our making and using visible absorption measurements experiment. In this laboratory, we're going to be using a spectrophotometer to obtain a visible spectrum of an absorbing species. We're also going to determine the analytical wavelength of an absorbing species. We're going to construct a Beer's law plot using absorbance readings of an absorbing species. And we're going to use our Beer's law plot to determine the concentration of an absorbing species solution with an unknown concentration. Let's get started. We first need to prepare our cuvettes to be read in the spectrophotometer. Today we're going to be using a 1 molar blue food dye stock solution. We also have our unknown blue food dye solution, unknown number 2. First, we're going to add 1 milliliter of deionized water to a cuvette, and this will serve as our blank. We're then going to add 1 milliliter of our unknown blue food dye solution to another cuvette. Next, we're going to prepare our standard dilutions of the 1 molar blue food dye stock solution. To do this, we're going to start by transferring 25 milliliters of our stock solution into a 25 milliliter volumetric flask. We're then going to transfer our 25 milliliters of 1 molar blue food dye solution to a 50 milliliter volumetric flask that will serve for our 1:2 dilution. We'll then fill our 50 milliliter volumetric flask with deionized water. And we'll invert to mix several times. We're then going to transfer 25 milliliters of our 1:2 blue food dye dilution into a new 25 milliliter volumetric flask. We're then going to transfer our 25 milliliters of our 1:2 dilution into a new 50 ml volumetric flask to make our 1:4 dilution. We'll then fill our 50 ml volumetric flask to the line with deionized water. We'll then cap and invert to mix. We're next going to add 25 milliliters of our 1:4 dilution to a new 25 milliliter volumetric flask. And now we're going to transfer our 25 milliliters of our 1:4 dilution into a new 50 ml volumetric flask for our 1:8 dilution. And then we'll fill our 50 ml volumetric flask up to the line with deionized water. And we'll invert to mix. We're now going to add 1 milliliter of our 1 molar, our 1:2 dilution, our 1:4 dilution, and our 1:8 dilution to cuvettes. We'll add 1 milliliter of our 1 molar stock food dye to a cuvette. We'll add 1 milliliter of our 1:2 dilution. We'll add 1 milliliter of our 1:4 dilution to a cuvette. And finally, we'll add 1 milliliter of our 1:8 dilution to a cuvette. We have now prepared all of our cuvettes to be read in the spectrophotometer. We have our blank, our unknown blue food dye solution, our 1 molar stock blue food dye solution, our 1:2 dilution, our 1:4 dilution, and our 1:8 dilution. We're next going to go over setup and operation of our spectrophotometer. So the first thing you need to do is turn it on and allow it to initialize. Upon initialization, we will then add one of our colored solutions in a cuvette to the spectrophotometer. After loading our cuvette with our colored solution into the spectrophotometer, we're now going to determine our analytical wavelength. So the first thing we need to do is press Enter to select the Spec 200E Modern Interface option. We're then going to press to the right 3 times under Application so that we have the Scan option come up. We're then going to press down. We're going to measure absorbance, so leave it there. And then we're going to press down again. We're going to set our low wavelength to 350 nanometers. And we're going to set our high wavelength to 700 nanometers. We'll then press down and we'll hit Enter. And then we'll hit Enter again. And the instrument will scan for the analytical wavelength that we should be using for our colored solution. The instrument is then going to show a range of wavelengths and the absorbance for each of those wavelengths for our colored solution. So for our blue food dye solution, the peak wavelength is right here. So we need to move our green line over so that we set our analytical wavelength at that peak wavelength. The peak wavelength for our blue food dye solution is about 634 nanometers. We next need to blank our spectrophotometer using a deionized water sample. So we're going to remove our colored food dye sample, and we're going to insert a deionized water sample. And then we're going to select our Spec 200E Modern Interface option. We're going to leave our application as live display. And we're going to set our measurement mode to % transmittance. We're going to leave our measurement wavelength or our analytical wavelength as the wavelength we have already previously determined, which is 634 nanometers. And then we're going to hit Enter on Go. We next need to select the 0 option to set our blank to 100% transmittance.
Now you can see that we have 100% transmittance for our deionized water sample. We have now completed the setup of our spectrophotometer, and we can read our samples. The first sample that we will be reading is our 1 molar blue food dye sample. The percent transmittance of our 1 molar blue food dye is 39.8%. We will next read our 1:2 dilution. The percent transmittance for our 1:2 dilution of our blue food dye is 63.6%. We will next read our 1:4 dilution of our blue food dye sample. The percent transmittance of our 1:4 dilution is 79.8%. We will now read our 1:8 dilution. Our 1:8 dilution has a percent transmittance of 88.5%. The next sample that we will be reading is our unknown blue food dye sample. Our unknown blue food dye sample has a percent transmittance of 82.7%. We have now completed the wet lab portion of this experiment.
Okay, students, you now need to convert our percent transmittance readings to absorbance and then make a graph of absorbance versus concentration. You can then determine the concentration of our unknown food dye solution by using the equation of a line from your graph. This concludes our making and using visible absorption measurements experiment. Thank you for joining me for this laboratory.